Hello everyone. What we've got here is a Key Stage 5, so we're talking A-level and IB video, on the Calvin cycle as part of the light independent reaction of photosynthesis. Now, lower down the school, you're always taught that photosynthesis happens in the sunlight, that you need sunlight for it to happen. So almost like a catalyst, along with carbon dioxide and water to form glucose and oxygen, and that in darkness it doesn't happen. Now, strictly that is not the case. There is a whole stage of photosynthesis that can happen in the absence of light, and that's what we're going to look at. Just 4 quick key points before we come to the Calvin cycle, named after clearly the scientist who predominantly did the most work on this. So 4 key points. First of all, 1, we're not needing light for this to happen. That's the first thing to note. Quite clearly, as the name suggests, the light independent. So that's one thing we must state from the get-go. But we can use light indirectly, although this predominantly does happen in darkness. Now, when we get sunlight and the plant is photosynthesizing, what we're producing actually isn't really a whole load of glucose to begin with. We're producing intermediate compounds, and it's those that can be made into glucose, amino acids, and other organic compounds. Now, by organic, I mean carbon-containing. And finally, that this process that I'm about to talk through takes place in the stroma of the chloroplast. It's a bit like the cytoplasm in a cell in a way. So you've got all the thylakoid membranes that are stacked from the grana, but we've got the kind of outer part that looks a bit like a cytoplasm. But in this organelle, we call it the stroma, and that's where this is going to take place. Now, for the Calvin cycle, what we're going to do is start with one of those intermediate compounds. We're going to start with a 5-carbon molecule. 3, 4, 5. So I'm just going to draw a 5-carbon molecule there. And what we've got here is one molecule. So I'll do one molecule, and it's got 5 carbons. We're just going to make a note. And this is known as ribulose bisphosphate. So I'll just put the name in place here. So that's ribulose bisphosphate. Now you can see that's quite a chunky name, so often it gets shortened, and it gets shortened to RuBP. So if you see RuBP in textbooks or on the internet, that's what we're referring to, ribulose bisphosphate. And it's a 5-carbon molecule. Now, what we have, if we were to draw this as a loop, let's say, is this molecule, because we're going to draw this as a cycle, put a little arrow there, it's essentially going to join with this here. And this molecule here, this is a molecule of carbon dioxide. Now what we have is carbon dioxide entering the chloroplast by diffusion, and it passes into the stroma. And this whole stage, this whole portion that I'm referring to now, is known as carbon fixation. We're fixing that carbon essentially. So this is all called carbon fixation. So carbon dioxide has entered the chloroplast by diffusion, passes into the stroma, and what we're going to do is get that carbon to essentially react with the ribulose bisphosphate, and that is catalyzed by an enzyme. And the enzyme we're going to use is called Rubisco. So we're going to use an enzyme called Rubisco as part of carbon fixation. Its real name is ribulose bisphosphate carboxylase, but we often shorten it to Rubisco. Now we have large quantities of that to maximize this process, because this step really is quite slow. So we're going to have a large quantity. This is a slow process here of carbon fixation. Often it's a rate limiting step, in fact. So what we end up with, when we've got that 5-carbon molecule with this one, would be 6 carbons. But rather than a 6-carbon molecule, what we actually have now are 2 molecules each one with 3 carbons in. So I'm just going to colour in these just to make it a little bit clearer, in fact, as we're going through. So we've essentially, if you just, from a simple perspective, if you just add the 5 carbons to the 1 carbon in carbon dioxide, that quite simply creates 6 carbons. But we have it as 2 3 carbon molecules. Now this molecule is known as glycerate 3-phosphate. So this is called glycerate 3-phosphate. It's sometimes also called 3-phosphoglycerate, but glycerate 3-phosphate is typically what you'd find. So that whole stage there is called carbon fixation. Now, this is where things turn a little interesting, because from that, what we now have, if we continue this process, is the formation of, again, another molecule, but this time something slightly different, and we produce 2 molecules of it. So it looks very similar. We've got 2 lots of a 3 carbon molecule. So we've gone from one chemical to another completely different one. So this one, or these 2 3 carbon molecules here, are known as triose phosphate. Now we've got 2 molecules, so we can write that down. We've got 2 molecules here, 2 mole, and each one has 3 carbons in it. Now, at that point, we've had to do something. To get glycerate 3-phosphate into triose phosphate, we've had to actually break down ATP. Now I'm just going to shrink the screen because I'm going to add this on as a sort of side bit to this loop that we're forming. I'll use a different colour just to make it a little bit clearer. So in the process of going from glycerate 3-phosphate to triose phosphate, so what we're referring to is this kind of stage here. At that point there, we've had 2 things happen. We've actually broken down ATP into ADP and a P, little I for inorganic. So that I there, P with an I, is an inorganic phosphate. We've broken down ATP, adenosine triphosphate, into ADP. So we've had to use a bit of energy for that. But we've also, if we were to do a balanced equation, have had 2 NADPH. And in other photosynthesis videos I've explained about what NADPH is. So you could refer to that one. And we have that going to 2 NADP plus. So we've had to basically use energy, but we've taken the hydrogen from the NADPH, essentially, or reduced NADP as it's called, and we've almost donated it to the glycerate 3-phosphate. So the NADPH has been oxidized, and the glycerate 3-phosphate, because it's gained the hydrogen, has been reduced. So glycerate 3-phosphate has been reduced to form triose phosphate. It's accepted that H+. Now once we've got triose phosphate, we can actually then almost rejoin this loop back up to RuBP. But I need to explain about what's going to happen in this process. Because when we form this loop, not all of this triose phosphate is essentially going to go back to being RuBP. Now, 5/6 of all of the triose phosphate must be used to regenerate RuBP. So we'll just make a note of that there, because that's a really key thing. That 5/6 1/6 of the triose phosphate is used to regenerate the RuBP. So you might be thinking, it seems a bit counterproductive. We've gone through this whole cycle and 5/6 of it we're just simply using to regenerate what we started with. But it's the 1/6, it's the 1/6 that's the essential component. So 1/6 of it is used to make glucose phosphate. Now that's a one molecule, it's just one molecule of that, but with 6 carbons in. So at this point here, just draw another little arrow. And whilst we had 5/6 of triose phosphate reforming or regenerated RuBP, what we've essentially got here is 1/6 used to make what's called glucose phosphate. Now, ATP and NADPH are used again in this process. Now, the condensation of many glucose phosphate molecules in the stroma together actually forms starch. So, quite simply, we said lower down in school you learn that glucose is stored as starch because it's insoluble, it's more compact for storage, and what we do to get that is essentially the condensation of many glucose phosphate molecules. So essentially we're going to use this glucose phosphate to make our glucose. We're going to get our glucose from this glucose phosphate. And this ultimately is the Calvin cycle. But one thing we need to just add to this here is that also in this stage ATP is being broken down into ADP and this inorganic phosphate. So just a reminder, I mean I've just said it there, but we are using ATP for this process to happen. Now, what's interesting is that I said 5/6 and 1/6. Now, chemically, you don't really deal with those kind of numbers. So what we do is we imagine we have 3 lots of RuBP. So if you work this through with me, I'll draw or make a note of this really at the top of the screen just here. So if you have 3 lots of RuBP, essentially that would give 6 molecules of triose phosphate. So that would give 6 lots of, if I just put TP, if I underline RuBP and TP, that will give 6 lots of triose phosphate. Because if you think we had 1 molecule here of RuBP producing 2 molecules, but each with 3 carbons, so we've gone from 1 to 2. So if we had 3 lots of RuBP, ratio-wise, we'd have 6 lots of triose phosphate. Ultimately, because the 2 molecules of glycerate 3-phosphate just reforms essentially 2 molecules of triose phosphate. So ultimately, 1 molecule of RuBP, 2 molecules of triose phosphate, 3 molecules of RuBP, 6 molecules of triose phosphate. So now we can say that instead of talking about 1/6 and 5/6, we can say that 1 of the 6 molecules goes to make glucose phosphate, and 5 of the 6 molecules goes into the regeneration of RuBP. So rather than talk about 1/6 and 5/6, we can say 1 molecule and 5 molecules. And this bit here is just really important to know because it's just a neater way to wrap up the numbers here. So that essentially is the Calvin cycle. We start with a molecule of RuBP, 5 carbons. In the process of carbon fixation using an enzyme called Rubisco, we get the addition of the carbon dioxide, or specifically that carbon, that 1 carbon, to form 2 lots of 3 carbon molecules of glycerate 3-phosphate. We use ATP and we oxidize NADPH to form 2 molecules of triose phosphate, and of that triose phosphate, 1/6 is going to be used to make glucose phosphate, and 5, 6 are going to be used to regenerate RuBP using ATP in the process. So I hope all that helps, and I'm going to be making a few more videos on the light independent reaction, just a few more specific details as a little added bonus. Okay, hope that helps.